Enzymes as biological catalysts. At the end of the 19th century, a debate was raging as to whether or not the process of ethanol formation required the presence of intact yeast cells. On one side of the debate was the organic chemist Justus von Liebig, who argued that the reactions of fermentation that produced the alcohol were no different from the types of organic reactions he had been studying in the test tube. On the other hand was the biologist Louis Pasteur, who argued that the process of fermentation could only occur within the confines of an intact, highly organized living cell. In 1897, 2 years after Pasteur's death, Hans Büchner, a bacteriologist, and his brother Edward, a chemist, were preparing yeast juice, an extract made by grinding yeast cells with sand grains and then filtering the mixture through filter paper. They wanted to preserve the yeast juice for later use. After trying to preserve the extract with antiseptic and failing, they attempted to protect the preparation from spoilage by adding sugar, the same process used to preserve jams and jellies. Instead of preserving the solution, the yeast juice produced gas from the sugar and it bubbled continuously for days. After further analysis, Edward discovered that fermentation was producing ethanol and bubbles of carbon dioxide. Buscher had shown that fermentation did not require the presence of intact cells. It was soon found, however, that fermentation was highly different from the types of reactions carried out by organic chemists. Fermentation required the presence of unique set of catalysts that had no counterpart in the non-living world. These catalysts were called enzymes. Enzymes are the mediators of metabolism, responsible for virtually every reaction that occurs in a cell. Without enzymes, metabolic reactions could proceed as so slowly as to be imperceptible. The first evidence that enzymes are proteins was obtained in 1926 by James Sumner of Cornell University when he crystallized the enzyme urease from jack beans and determined its composition. Though this finding was not greeted with much positive acclaim at the time, several other enzymes were soon shown to be proteins, and it became accepted for the next few decades that all biological catalysts were proteins. Eventually, it became evident that certain biological reactions are catalyzed by RNA molecules. For the sake of clarity, the term enzyme is still generally reserved for protein catalysts, while the term ribozyme is used for an RNA catalysts. Even though enzymes are proteins, many of them are conjugated proteins. That is, they contain non-protein components called cofactors, which may be inorganic, metallic, or organic coenzymes. Vitamins and their derivatives often function as coenzymes. When present, cofactors are important participants in the functioning of the enzyme, often carrying out activities for which amino acids are not suited. In myoglobin, for example, as discussed in the previous chapter, the iron atom of the heme group is the site where oxygen is bound and stored until required for further cellular metabolism. The properties of enzymes, as is true for of all catalysts, enzymes exhibit the following properties. They are required only in small amounts. They are not altered irreversibly during the course of the reaction, and therefore each enzyme molecule can participate repeatedly in individual reactions. They have no effect on the thermodynamic properties of the reaction. They have no effect on the thermodynamics of the reaction. This last point is particularly important. Enzymes do not supply energy for a chemical reaction and therefore do not determine whether a reaction is thermodynamically favorable or unfavorable. Similarly, enzymes do not determine the ratio of products to reactants at equilibrium. These are inherent properties of the reacting chemicals. As catalysts, enzymes can only accelerate the rate at which the favorable chemical reaction proceeds. There is no necessary relationship between the magnitude of the delta G for a particular reaction and the rate at which this reaction takes place. The magnitude of delta G informs us only of the difference in free energy between the beginning state and equilibrium. It is totally independent of the pathway or the time it takes to reach equilibrium. The oxidation of glucose, for example, is a very favorable process as can be determined by the amount of energy released during its combustion. However, glucose crystals can be left out in room air virtually indefinitely without a noticeable conversion into less energetic materials. Glucose, in other words, is kinetically stable even if it is thermodynamically unstable. So, glucose is kinetically stable, as we can see. It's kinetically stable even if it is thermodynamically unstable. Even if the sugar were to be dissolved, as long as the solution were kept sterile, it would not rapidly deteriorate. However, if one were to add a few bacteria within a short period of time, the sugar would be taken up by the cells and enzymatically degraded. Enzymes are remarkably adept catalysts employed by organic chemists in the lab, such as acid, metallic, platinum, and magnesium, generally accelerate reactions in 100 to 1000 times over the non-catalyzed rate. In contrast, enzymes typically increase the velocity of reaction 10 raised to 8 to 10 raised to 13 fold or greater. 10 raised to 8 or 10 raised to 13 or fold or greater. On the basis of these numbers, enzyme can accomplish in 1 second what would require from 3 years to 300,000 years if the enzymes were absent. Even more remarkably, they accomplish this feat at the mild temperature and pH found within the cell. In addition, unlike the inorganic catalysts used by chemists, enzymes are highly specific in the reactions they can bind and the reaction they can catalyze. The reactants bound by an enzyme are called substrates. If, for example, the enzyme hexokinase is present in solution with 100 low molecular weight compounds in addition to its substrate, glucose, only the glucose molecule will be recognized by the enzyme and undergo reaction. For all practical purpose, the other compounds may as well be absent. This type of specificity, whether between enzymes and substrates or other type of proteins and the substances that they bind, is crucial for maintaining the order required to sustain life. In addition to their high level of activity and specificity, enzymes act as metabolic traffic directors in the sense that enzyme catalyzed reactions are very orderly. The only products formed are the appropriate ones. This is very important because the formation of chemical byproducts would rapidly take its toll on the life of a fragile cell. Finally, unlike other catalysts, the activity of enzymes can be regulated to meet the particular needs of the cell at a particular time. As will be evident in this chapter, the remainder of the text, the enzymes in a cell are a truly wondrous collection of miniature molecular machines. So, first, let's learn about overcoming the activation energy barrier. How are enzymes able to accomplish such effective catalysis? The first question to consider is why thermodynamically favorable reactions do not proceed on their own at relatively rapid rates in the absence of enzymes. Even ATP, whose hydrolysis is so favorable, is stable in a cell until broken down in a controlled enzymatic reaction. If this weren't the case, ATP would have little biological use because that just deteriorates when it's idle. So, chemical transformations require that certain covalent bonds be broken within the reactants. For this to occur, the reactants must contain sufficient kinetic energy of motion, that they overcome a barrier called the activation energy. This is expressed diagrammatically, where the activation energy is represented by the height of the curves. The reactants in a chemical reaction are often compared to an object resting on top of a cliff, ready to plunge into the bottom. If left to itself, the object in all likelihood would remain there indefinitely. However, if someone were to come along and provide the object with sufficient energy to overcome the friction or other small barrier in its way and cause it to reach the edge of the cliff, it would spontaneously drop to the bottom. The object has the potential to fall to a lower energy state once the kinetic barrier have been removed. In a solution at room temperature, molecules exist in a state of random movement, each possessing a certain amount of kinetic energy at a given instant. Among a population of molecules, their energy is distributed in a bell-shaped curve, some having very little energy and others much more. Molecules of higher energy, that is, activated molecules, remain as such only for a brief time, losing their excess energy to other molecules by collision. Let us consider a reaction in which one reactant molecule is split into two product molecules. If a given reactant molecule acquires sufficient energy to overcome the activation barrier, then the possibility exists that it will split into the two product molecules. The rate of the reaction depends on the number of reactant molecules that contain the necessary kinetic energy at any given time. One way to increase the reaction rate is to increase the energy of the reactants. This is most readily done in the laboratory by heating the reaction mixture. In contrast, trying heat to an enzyme-mediated reaction leads to the rapid inactivation of the enzyme due to its denaturation. Reactants, when they are at the crest of the energy hump and ready to be converted to products, are said to be at the transition state. So, course of reaction will be represented on the x-axis. Free energy will be represented on the y-axis. Final state will be there. Then initial state is happening. So reactants have a particular activation energy barrier. Then they cross that transition state, activated complex between glucose and ATP. Then EA of uncatalyzed activation energy of uncatalyzed reaction in forward direction. Activation energy of enzyme catalyzed reaction in forward direction. Course of reaction. Free energy, glucose plus ATP. Transition state, activated complex between glucose and ATP. Enzyme activation energy of uncatalyzed reaction in forward reaction. Activation energy of enzyme catalyzed reaction in forward direction. Products are there. After reaching that transition state, they transform to products. The effect of lowering activation energy on the rate of reaction. The bell-shaped curves indicate the energy content of a population of molecules present in a reaction mixture at two different temperatures. The number of reactant molecules containing sufficient energy to undergo reaction is increased by either heating the mixture or adding an enzyme catalyst. Heat increases the rate of reaction by increasing the energy content of the molecule, while the enzyme does so by lowering the activation energy required for the reaction to occur. Number of molecules with a particular energy will be there. Energy is created, and first we will be providing 25 degrees Celsius. So minimal energy required for molecules for catalyzed reaction will be there. So minimal energy required for molecules for uncatalyzed reaction will be there. So molecules capable of reaction in presence of enzyme catalysts at lower temperature is shown, and molecules capable of reaction in elevated temperature without catalyst. Molecules capable of reaction in lower temperature, no catalyst is also there. So at this point, transition state, we are talking about the transition state. At the transition state point, the reactants have formed a fleeting activated complex in which bonds are being formed and broken. We can illustrate the nature of the transition state structure by examining the interconversion of D and L stereoisomers of proline, a reaction catalyzed by the bacterial enzyme proline racemase. So D-proline will be there and transition state is happening and L-proline is created. This reaction proceeds in either direction through the loss of a proton from the alpha carbon of the proline molecule. As a result, the transition state structure contains a negatively charged carbanion in which the three bonds formed by the carbon atom are all in the same plane. Unlike the difference in standard free energy for a reaction, the activation energy required to reach the transition state is not a fixed value. but varies with the particular reaction mechanism utilized. Enzymes catalyze reactions by decreasing the magnitude of the activation energy barrier. Consequently, unlike catalysis by heat, catalyze reactions by decreasing the magnitude of the activation energy barrier. Consequently, unlike catalysis by heat, enzymes cause their substrate to be very reactive without having to be raised to particularly high energy levels. A comparison between the percentage of molecules able to react in an enzyme catalyzed reaction versus a non-catalyzed reaction can be graphically represented. Enzymes are able to lower activation energies by binding more tightly to the transition state than to the reactants, which stabilize this activated complex, thereby decreasing its energy. As a result, differences in the geometry and bonding properties between the reactants and the transition state are a focus of research in enzymatic mechanisms. The importance of the transition state can be demonstrated in numerous ways. For example, compounds that resemble the transition state of a reaction tend to be very effective inhibitors of that reaction because they are able to bind so tightly to the catalytic region of the enzyme. Antibodies normally do not behave like enzymes but rather simply bind to molecules with high affinity. However, antibodies that bind to a compound that resembles a transition state for a reaction are often able to act like enzymes and catalyze the breakdown of that compound. Antibodies normally do not behave like enzymes but rather simply bind to molecules with high affinity. However, antibodies that bind to a compound that resembles a transition state for a reaction are often able to act like enzymes and catalyze the breakdown of that compound. As the transition state is converted to products, the affinity of the enzyme for the bound molecule decreases and the products are expelled. Now let's learn about the active site. As catalysts, enzymes accelerate bond breaking and bond forming process. To accomplish this task, enzymes become intimately associated with the activities that are taking place along the reactants. Enzymes do this by forming a complex with reactants called enzyme substrate complex. An enzyme substrate complex can be shown, and enzyme will be there, ADP and PEP will be there. Enzyme substrate complex is formed and they just release ATP and pyruvate are released. Formation of an enzyme substrate complex. Schematic drawing of the reaction catalyzed by pyruvate kinase, in which the two substrates phosphoenol pyruvate and ADP bind with the enzyme to form an enzyme substrate complex which leads to the formation of the products pyruvate and ATP. Enzymes and substrate is non-covalent, so many examples are non-transient covalent bond is formed. That part of the enzyme molecule that is directly involved in binding the substrate is termed as an active site. The active site and the substrate have complementary shapes, enabling them to bind together with a high degree of precision. The binding of a substrate to enzyme is accomplished by the same types of non-covalent interactions like ionic bonds, hydrogen bonds, hydrophobic interactions that determine the structure of the protein itself. For example, the enzyme contains a number of positively charged residues strategically located to bind negatively charged atoms of the substrate. In addition to binding the substrate, the active site contains a particular array of amino acid side chains that influence the substrate and lower the activation energy of the reaction. The importance of individual side chains of the active site can be evaluated by site-directed mutagenesis technique, in which one particular amino acid is replaced by another amino acid having different properties. So site-directed mutagenesis will be used here. The active site is typically buried in a cleft or portion that leads from the aqueous surroundings into the depths of the protein. When a substrate enters the active site cleft, it typically gives up its bond water molecules dissolution and enters a hydrophobic environment within the enzyme. The reactivity of the active site chains may be much greater. In its protected environment as compared to the aqueous solvent of the cell. The amino acids that make up the active site are usually situated at distant points along the length of the extended polypeptide chain, but they are brought into close proximity to one another as the polypeptide folds into its final tertiary structure. The structure of the active site accounts not only for the catalytic activity of the enzyme but also for its specificity. As noted above, most enzymes are capable of binding only one or a small number of closely related biological molecules. Tubulose biphosphate carboxylase oxygenase, showing the various sites of reaction between the bond substrates RUBP and CO2 and certain amino acid side chains of the enzyme is an example. In addition to determining the substrate binding properties of the active site, this non-covalent interaction alters the properties of the substrate in ways that accelerate its conversion to products. An electron density map of the active site of a viral thermothymine kinase with the substrate deoxyrimidine is shown. Now let's learn the mechanism of enzyme catalysis. How is it that an enzyme can cause a reaction to occur hundreds of times a second when that same reaction might occur at undetectable rates in the enzyme's absence? The answer lies in the formation of the enzyme substrate complex, which allows the substrate to be taken out of solution and held onto the surface of the large catalytic molecule. Once there, the physical and chemical properties of the substrate can be affected in a number of ways, several of which are described in the following sections. First is the substrate orientation. Suppose you were to place a handful of nuts and bolts into a bag and shake the bag for 5 minutes. It is very unlikely that any of the bolts would have a nut firmly attached to its end and when you were finished. In contrast, if you were to pick up a bolt in one hand and a nut in the other, you would rapidly guide the bolt into the nut. By holding the nut and bolt in the proper orientation, you have greatly increased the entropy of the system. Enzymes lower the entropy of their substrates in a similar manner. Substrates bound to the surface of the enzymes are brought very close together in precisely the correct orientation to undergo reaction. In contrast, when reactants are present in solution, they are free to undergo translation and rotational movements, and even those possessing sufficient energy do not necessarily undergo a collision that results in the formation of a transition state complex. So, changing substrate reactivity. Enzymes are composed of amino acids having a variety of different types of side chains, from fully charged to highly nonpolar. When a substrate is bound to the surface of an enzyme, the distribution of the electrons within that substrate molecule is influenced by the neighboring side chains of the enzyme. This influence increases the activity of the substrate and catalyzes the transition state complex formed during the reaction. These effects are accomplished without the input of external energy, such as heat. There are several general mechanisms whereby the reactivity of substrates is increased by association with enzymes. Basically, these mechanisms are similar to those characterized by organic chemists studying the mechanisms of organic reactions in a test tube. For example, the rate of reactions can be greatly affected by changing the pH. Although enzymes cannot change the pH of their medium, they do contain numerous amino acids with acidic or basic side chains. These groups are capable of donating or accepting protons to and from the substrate, thereby altering the electrostatic character of the substrate, making it more reactive. The active sites of many enzymes contain side chains with a partial positive or negative charge. Such groups are capable of interacting with the substrate to alter its electrostatic properties and hence its reactivity. Such groups are also capable of reacting with a substrate to produce a temporary covalent enzyme substrate linkage. Chymotrypsin. Chymotrypsin, an enzyme that digests food proteins within the small intestine, acts in this way. A series of reactions that take place as chymotrypsin hydrolyzes a peptide bond in a substrate protein is shown, divided into two steps. This process, in the first step, the electronegative oxygen atom of the side chain of serine of the enzyme attacks a carbon atom of the substrate. As a result, the peptide bond of the substrate is hydrolyzed, and a covalent bond is formed between the serine and the substrate, displacing the remainder of the substrate as one of the products. As discussed in the figure, ligand, the ability of serine to carry out this reaction depends on a nearby histidine residue which attracts the proton of serine's hydroxyl group, increasing the nucleophilic power of the group's oxygen atom. Enzymes are also adept at utilizing water molecules in the reactions they catalyze. In the second step, the covalent bond between enzyme and substrate is cleaved by a water molecule, returning the enzyme to its original unbound state and releasing the remainder of the substrate as the second product. Although amino acid side chains can engage in a variety of reactions, they are not well suited to donate or accept electrons. As we see in the following section, the transfer of electrons is the central event. So substrate aligned enzyme is there and the substrate aqueous charged region, substrate stress. Three mechanisms by which enzyme accelerate reactions: maintaining precise substrate orientation, changing substrate reactivity, altering its electrostatic configuration, exerting physical stress on bonds in the substrate to be broken. To catalyze these reactions, enzymes contain cofactors, metal ions or coenzymes that increase the reactivity of substances by removing or donating electrons. In a number of cases, enzymes create their own electron accepting cofactor by chemically modifying one of the amino acid residues situated in their active site. Then the third point is the inducing strain in the substrate. Although the active site of an enzyme may be complementary to its substrate, various studies reveal a shift in the relative position of certain of the atoms of an enzyme once the substrate has bound. In many cases, the conformation shifts so that the complementary fit between the enzyme and reactants is improved, and an induced fit and the proper reaction groups of the enzyme move into place. An example of induced fit is given. This type of movements within an enzyme molecule provide a good example of protein acting as a molecular machine as these conformational changes occur. Mechanical work is performed, allowing to the enzyme to exert a physical force on certain bonds within a substrate molecule. This has an effect of destabilizing the substrate, causing it to adopt a transition state in which the strain is relieved. Studying conformational changes and catalytic intermediates. To fully understand the mechanism by which an enzyme catalyzes a particular reaction, it is necessary to describe the various changes in atomic and electronic structure in both the enzyme and the substrate as the reaction proceeds. We saw in the last chapter how X-ray crystallographic techniques can reveal details of the structure of large enzyme molecule. Since 40 to 60% of the volume of a typical protein crystal consists of a trapped solvent, most crystallized enzymes retain a high level of enzymatic activity. It should be possible, therefore, to use X-ray crystallographic techniques to study reaction mechanisms. There is one major limitation, namely time. In a standard crystallographic study, enzyme crystals must be subjected to an X-ray beam for a period of hours or days while the necessary data are collected. The portrait that emerges from such studies capture the structure of the molecule averaged over time. Recent innovations, however, were made to possible to use X-ray crystallographic techniques to observe the flaring structural changes that take place in the active site while an enzyme is catalyzing a single reaction cycle. This approach, which is called time-resolved crystallography, can include use of ultra-high intensity X-ray beams generated by a synchrotron, an instrument used by nuclear physicians to study subatomic particles. This can put X-ray exposure period to a matter of picosecond, which is the same time scale required for an enzyme to catalyze a single chemical transformation. Cooling of the enzyme crystals to temperature within 20 to 40 degrees of absolute zero, which slows the reaction, by a factor as high as 10 billion, greatly increasing lifetime of transient intermediates. Use of techniques to simultaneously trigger a reaction throughout an entire crystal so that all of the enzyme molecules in the crystal are in the same stage of the reaction at the same time. For example, in the case of reaction in which ATP is a substrate, the enzyme crystals can be infiltrated with ATP molecules that have been made in freeze non-reactive by linking them to an inert group, nitrophenyl group, in a photosensitive bond. When the crystals are exposed to a brief pulse of light, all of the caged ATP molecules are released, initiating the reaction simultaneously in the active sites throughout the crystal. Use of enzyme bearing site-directed mutations that impose kinetic barriers at specific stages of the reaction, leading to an increased lifetime for particular intermediates. Then there's the determination of the structure at ultra high atomic resolution, in which allows visualization of individual hydrogens that may be present in hydrogen bonds or associated with acidic groups in the protein. The presence or absence of bound water molecules and precise confirmation of catalytic side chains and the subtle strain that appears in parts of the substrate during catalysis. The remarkable detail that can be extracted from this high resolution images is illustrated by the hydrogen bond. By combining these techniques, researchers have been able to determine the three-dimensional structures of enzymes at successive stages during a single catalysis reaction. And these individual snapshots are put together in sequence. They produce a movie showing the various catalytic intermediates that appear and disappear in the reaction proceeds. An example of the data is seen. Well, the spotlight has been focused on the conformational changes that occur as an enzyme transforms its substrates into products. Considerable attention has also been paid to in recent years to the conformation changes that occur within the protein itself. Proteins are dynamic molecules that are capable of built-in or intrinsic motion that can be important in functional relevance. This motion allows entry of substrate to the enzyme's active site. Catalysis of this type of intrinsic motion by various experimental techniques has suggested that proteins, even in the absence of substrate, are capable of many of the same movements that can be detected during the protein's catalytic cycle. If true, this suggests that evolution has selected for protein structure that are capable of intrinsic motions that can be useful for potential functions that a protein might serve. Rather than inducing a specific conformational change, the substrates might simply be waiting for a protein to assume a conformation to which it can effectively bind. Now, let's learn about the enzyme kinetics. Enzymes vary greatly in their ability to catalyze reactions. The catalytic activity of an enzyme is revealed by studying its kinetics, that is, the rate at which it catalyzes a reaction under various experimental conditions. In 1913, Leonor Michaelis and Maud Menton reported on the mathematical relationship between substrate concentration and the velocity of enzyme reactions as mentioned by the amount of product formed or substrate consumed in a given amount of time. This relationship can be expressed by an equation that generates a hyperbola. Rather than considering the theoretical aspects of enzyme kinetics, we can obtain the same curve in a practical manner as it is done for each enzyme studied. To determine the velocity of the reaction, an incubation mixture is set up at the desired temperature containing all the ingredients required except one, which, when added, initiates the reaction. If at the time the reaction begins, no product is present in the mixture, then the amount of product that appear over time provides a measure of the velocity of the reaction. There are compiling there are complicating factors in this procedure. If the incubation time is too great, the substrate concentration becomes measurably reduced. In addition, as product appears, it can be converted back to the substrate by reverse reaction, which is also catalyzed by the enzyme. Ideally, what we want to determine is the initial velocity, that is, the velocity at the instant when no product has yet been formed. To accurately measure the initial reaction velocity, In addition, as product appears, it can be converted back to substrate by the reverse reaction, which is also catalyzed by the enzyme. Ideally, what we want to determine is the initial velocity, that is, the velocity at the instant when no product has been yet formed. To accurately measure the initial reaction velocity, brief incubation times, and sensitive measuring techniques are employed. To generate a curve such as the initial velocity is determined for a series of incubation mixtures that contain the same amount of enzyme but an increasing concentration of substrate. It is evident from this curve that the initial velocity varies markedly with substrate concentration. At low substrate concentrations, enzyme molecules are subjected to relatively few collisions with substrate in a given amount of time. Consequently, the enzyme has idle time, that is, substrate molecules are rate limiting. At high substrate concentrations, enzymes are colliding with substrate molecules more rapidly than they can be converted to product. Thus, at high substrate concentrations, individual enzymes molecules are working at their maximum capacity. That is, enzyme molecules are rate limiting. Thus, as a greater and greater concentration of substrate is present in the reaction mixture, the enzyme approaches a state of saturation. This initial velocity at this theoretical saturation point is termed the maximum velocity. Since each enzyme molecule is only able to catalyze a certain number of reactions in a given amount of time, the velocity of the reaction, typically expressed as moles of product formed per second, approaches a maximal rate as the substrate concentration increases. The substrate concentration at which the reaction is at half maximal velocity is called the Michaelis constant, or Km. The substrate concentration at which the reaction is at half maximal velocity is called Michaelis constant, or Km. To generate a graph, the initial velocity is determined for a series of incubation mixtures that contain the same amount of enzyme but an increasing concentration of substrate. The simplest measure of the catalytic activity of an enzyme is provided by its turnover number, which can be calculated from the Vmax. The turnover number, or catalytic constant, it is also called turnover number, catalytic constant, is the maximum number of molecules of substrate that can be converted to product by one enzyme molecule per unit time. The maximum number of molecules of substrate that can be converted into product by one enzyme molecule per unit time. A turnover number per second of 1 to 10 raised to 3 is a typical for enzymes, although values as great as 10 raised to 6 are not. It is apparent from these values that a few molecules of enzymes can rapidly convert a large number of substrate molecules into products. The value of Vmax is only one useful term obtained from a plot, such as explained earlier. Another is the Michaelis constant Km, which is equal to the substrate concentration when the reaction velocity is 1/2 of Vmax. As its name implies, the Michaelis constant Km is constant for a given enzyme and this independent of substrate or enzyme concentration. The relationship between Vmax and Km is best appreciated by considering the Michaelis-Menten equation, which can be used to generate a plot given V is equal to Vmax delta S by V is equal to Vmax concentration of S by concentration of S plus Km. V is equal to Vmax into concentration of S by concentration of S plus Km. Okay. According to this equation, when the substrate concentration S is at a value equivalent to Km, then the velocity of the reaction V becomes equal to Vmax by 2. For example, we will be, calculating S as is equal to Km. S is about equal to Km. So, Vmax by 2. V is equal to Vmax by 2. V is equal to Vmax by 2. That is what is going to happen. For 1/2 of the maximal velocity, thus Km is equal to S where V is equal to Vmax by 2. When Km is equal to S, then V is equal to Vmax by 2. To generate a hyperbolic curve and make an accurate determination of the values for Vmax and Km, a considerable number of points must be plotted. An easier and more accurate description is gained by plotting the reciprocals of the velocity and substrate concentration against one another as formulated by Hans, Lineweaver and Dianbrook. When this is done, the hyperbola becomes a straight line whose x-intercept is equal to minus 1 by km, y-intercept is equal to 1 by Vmax, and slope is equal to km by Vmax. So this graph is something which is very important. So x-intercept will be equal to minus 1 by km, and y-intercept is nothing else but 1 by Vmax. Slope will be Km by Vmax. Okay. The values of Km and Vmax are therefore readily determined by extrapolation of the line drawn from a relatively few points. In most cases, the value of Km provides a measure of the affinity of the enzyme for the substrate. But Km represents affinity. Km is equal to affinity of the enzyme for the substrate, affinity of enzyme for substrate. That is what is represented by Km. So the x-intercept is minus 1 by Km, y-intercept is 1 by Vmax, slope is Km by Vmax. Then we are getting a straight line of a Lineweaver-Burke plot of the reciprocal of velocity and substrate concentration from which the values for the Vmax and Km are readily calculated. 1 by V will be representing on the y-axis, and 1 by S is represented on the x-axis, 1 by V and 1 by concentration of S, 1 by Vmax slope, and slope is equal to Km by Vmax. X-intercept is equal to 1 by Km minus 1 by Km. Y is equal to 1 by Vmax. Slope is equal to Km by Vmax. Km is equal to affinity of enzyme for substrate. Enzyme of that substance. The Km for most enzyme ranges between 10 raised to minus 1 and 10 raised to minus 7 with a typical value about 10 raised to minus 4 m. Other factors that strongly influence enzyme kinetics are the pH and temperature of the incubation medium. Each enzyme has an optimal pH and temperature at which it operates at maximum activity. The influence of temperature on enzyme activity is illustrated by a striking effect of refrigeration by showing the growth of microorganisms. Changes in pH affect the ionic properties of the substrate and the enzymes as well as enzyme conformation. At lower temperature, reaction rates with increasing temperature due to the increased energy of the reactants. At higher temperature, this positive effect is offset by enzyme denaturation. Enzyme inhibitors. Enzyme inhibitors are molecules that are able to bind to an enzyme and decrease its activity. Enzyme inhibitors are molecules that are able to bind to an enzyme and decrease its activity. The cell depends on inhibitors to regulate the activity of many of its enzymes. Biochemists use inhibitors to study the properties of enzymes, and many pharmaceutical companies produce enzyme inhibitors that act as drugs. Enzyme inhibitors can be divided into two types: reversible or irreversible. Reversible inhibitors are those that bind very tightly to an enzyme, often by forming a covalent bond to one of its amino acid residues. So, that is something which is termed as an irreversible inhibitor. Those inhibitors which bind very tightly to an enzyme, often by forming a covalent bond to one of its amino acid residues. A number of nerve gases such as organophosphate pesticides and dis diisopropyl phosphofluoridate fluoridate 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 and the organophosphate pesticides act as irreversible inhibitors of acetylcholinesterase, an enzyme that plays a crucial role in destroying acetylcholine, the neurotransmitter responsible for causing muscle contraction. When the enzyme inhibited, the muscle is stimulated continuously and remains in a state of permanent contraction. The antibiotic penicillin acts as an irreversible inhibitor for a key enzyme in the formation of the bacterial cell wall. Reversible inhibitors, on the other hand, bind only loosely to an enzyme and thus are readily displaced. For the sake of simplicity, we will discuss the simplest cases of reversible inhibitors, namely the competitive and uncompetitive type. So, competitive inhibitors are reversible inhibitors that compete with a substrate for access to the active site of an enzyme. Since the substrates have a complementarity structure to the active site to which they bind, competitive inhibitors must resemble the substrate to compete for the same active site but differ in a way that prevents them from being transformed into product. Analysis of the types of molecules that can compete with the substrate for a binding site on the enzyme provides insight into the structure of the active site and the nature of the interaction between a substrate and its enzymes. Competitive enzyme inhibitors forms the basis of action of many common drugs. Angiotensin converting enzyme is a proteolytic enzyme that acts on a 10 residue peptide angiotensin 1 to produce an 8 residue peptide angiotensin 2. Elevated levels of angiotensin 2 acts as a major risk factor in the development of the high blood pressure or hypertension. In the 1960s, John Wayne and his colleagues at the Eli Lilly company began to search for compounds that could inhibit ACE or angiotensin. Previous studies had found that the venom of Brazilian pit viper contained inhibitors of proteolytic enzymes, and it was found that one of the components of this venom, a peptide called teprotide, was a potent competitive inhibitor of angiotensinogen. Although teprotide was shown to lower blood pressure in a hypertensive patients, it was not a very useful drug because it had a peptide structure and thus was rapidly degraded if taken orally. Subsequent efforts to develop non-peptide inhibitors of the enzyme led researchers to synthesize a compound called captopril, which became the first useful anti-hypertensive drug that acted by binding to ACE. That is the captopril. Captopril. The effectiveness of a competitive inhibitor depends on its relative affinity for the enzyme. Regardless, competitive inhibition can be overcome if the substrate inhibitor ratio is great enough. In other words, if the number of collisions between the enzyme and inhibitor becomes insignificantly relative to those between the enzyme and its substrate, then the effect of the inhibitor becomes minimal. Given a sufficient substrate concentration, it remains theoretically possible to achieve the enzyme's maximum velocity even in the presence of the competitive inhibitor. Given a sufficient substrate concentration, it remains theoretically possible to achieve the enzyme's maximum velocity even in the presence of the competitive inhibitor. Substrate will be there, active site will be there, enzyme is there. So inhibitors are just actively competing with the substrate molecules for the access into the active site. Because of their molecular similarity, competitive inhibitors are able to compete with the substrate for a binding site on the enzyme. The effect of a competitive inhibitor depends on the relative concentrations of the inhibitor and substrate. Next is the non-competitive inhibition. The substrate and inhibitor do not compete for the same binding site. Generally, the inhibitor acts as a site other than the enzyme's active site. The level of inhibition depends only on the concentration of the inhibitor, and increasing the concentration of the substrate cannot overcome it. Since in the presence of the non-competitive inhibitor, a certain fraction of the enzyme molecules are necessarily inactive at a given instant, the maximal velocity of the population of enzyme molecules cannot be reached. Tripna Tipranavir is a potent non-competitive inhibitor of the protease produced by HIV when it infects a white blood cell. Unlike other inhibitors of this enzyme, such as ritonavir, tipranavir does not resemble the peptide substrate of the enzyme, nor does it compete with the substrate. The effects on the kinetics of enzymes in the presence of non-competitive and competitive inhibitors are given. In one case, the Vmax is lowered, and on the other, the Km is increased. In both types, the slope is increased relative to the uninhibited reaction. As we will see, cells utilize a version of uncompetitive inhibition to regulate the activity of key enzymes of metabolic pathways. So, when we plot a graph of concentration of S against V, we can get three lines which are just curved lines. First one is a Vmax by 2, that is the non-competitive inhibition. Then the competitive inhibition will be happening. Then uninhibited enzyme will be there, that is the most biggest curve. The effect of inhibitors on enzyme kinetics. The effect of both competitive and non-competitive inhibitors is shown. The effect of both competitive and non-competitive inhibitors is shown when the kinetics of the reaction are plotted as velocity of reaction versus substrate concentration or its reciprocal B. The uncompetitive inhibitor reduces Vmax without affecting Km, whereas the competitive inhibitor increases Km without affecting Vmax. Now, let's learn about the human perspective of this enzyme into our real life. That is the growing problem of antibiotic resistance. Not too long ago, it was widely believed that human health would no longer be threatened by serious bacterial infections. Bacterial diseases such as leprosy, pneumonia, gonorrhea, and dozens of others were being cured by administration of any one of a number of antibiotics, compounds that selectively kill bacteria without harming the human host in which they grow. This state of affairs has changed dramatically in the past couple of decades as pathogenic bacteria have become increasingly resistant to those wonder drugs, leading to the deaths of many people who would once have been successfully treated. Even tuberculosis, which had virtually disappeared from developed countries, has reemerged around the world in a form referred to as XDR, extremely drug resistant, that is virtually untreatable. It is feared that XDR or extremely drug resistant TB is on the verge of becoming a global major health crisis. To make matters worse, the pharmaceutical industry has dramatically cut resources devoted to the development of new antibiotics. This change of action by the drug industry is generally attributed to the lack of financial incentives. Antibiotics are taken only for a short period of time as opposed to drugs prescribed for chronic conditions such as diabetes or depression. Unlike other types of drugs that retain their effectiveness or efficacy, new antibiotics turn the risk of having a relatively short lifetime in the marketplace as bacteria become resistant to each successive product. The most effective antibiotics are held back from the widespread use and are kept instead as weapons of last resort when other drugs have failed. Ideas for the formation of non-profit institutions for the development of new antibiotics have been widely discussed. Here we will look at briefly at the mechanism of action of antibiotics, particularly those that target enzymes, which are the substances which are the most important part we are dealing with, and the development of bacterial resistance. Most antibiotics are derived from mutual products, natural products that are produced by microorganisms living in the soil and readily cultivated in the lab. Many classes of antibiotics are there. Targets in the bacterial cell on which these compounds act, and the mechanism by which bacteria are developed resistance to that class of compounds. We will first look at several of the main types of targets. Enzymes involved in the synthesis of the bacterial cell wall. Penicillin and its derivatives, methicillin, are structural analogues of the substrates of a family of transpeptidases that catalyze the final cross-linking reactions that give the cell wall its rigidity. If these reactions do not occur, a rigid cell wall fails to develop. Penicillin is an irreversible inhibitor of the transpeptidases. The antibiotic fits into the active site of these enzymes, forming a covalently bound complex that cannot be disloaded. The antibiotic vancomycin inhibits transpeptidases by binding to the peptide substrate of the transpeptidases rather than to the enzyme itself. Normally, the transpeptidase substrate terminates in a d-alanine dipeptide. To become resistant to vancomycin, a bacterial cell must synthesize an alternative terminus that doesn't bind to the drug, which is a roundabout process that requires the acquisition of several new enzymatic activities. As a result, vancomycin is the antibiotic to which bacteria have proven least able to develop resistance, and this is usually given as last resort when other antibiotics are failed. So, vancomycin is very important. Unfortunately, vancomycin resistant strains of several pathogenic bacteria, including Saccharomyces, Staphylococcus aureus, have emerged in recent years. Staphylococcus aureus is the common inhabitant of the skin and nasal passages. Although it is usually relatively harmless, this organism is the most frequent cause of life-threatening infections that develop in hospitalized persons who have open wounds or invasive tubes. For years, methicillin resistant Staphylococcus aureus strains, which are also resistant to many other antibiotics, have created havoc in hospital wards. Infections by with this one pathogen kill approximately 20,000 patients every year in the United States alone, which is greater than the number of deaths in the United States from AIDS. MRSA infections have also begun to appear in community settings such as high school gyms or children's daycare centers where there is a lot of contact between individuals. In many cases, vancomycin is the only drug that can stop these infections. Consequently, it was alarming to discover that MRSA can become resistant to vancomycin by acquiring a cluster of vancomycin resistant genes from another bacterium, which is also a common cause of hospital based infections. So far, not known vancomycin resistant MRSA have been able to gain a foothold in either a hospital or community setting, but it may only be a matter of time. For this reason, infectious disease specialists are urged hospitals to institutions better hygiene programs and to isolate patients at a first sign of infection. This practice have been proven to reduce the incidence of lethal infections where they have been put into place. Then the components of the system by which bacteria duplicate, transcribe and translate their genetic information. That is another point. Although bacterial and eukaryotic cells have a similar system of storing and utilizing genetic information, there are many differences between the two types of cells that pharmacologists can take advantage of. Streptomycin and the tetracyclines, for example, bind to bacterial ribosomes but not eukaryotic ribosomes. Quinolones such as ciprofloxacin, brand name Cipro, are a rare example of wholly synthetic antibiotics. They are not based on natural products. Quinolones inhibit the enzyme DNA gyrase, which is required for bacterial DNA replication. Enzymes that catalyze metabolic reactions, specifically in bacteria. Nearly all of the new antibiotics approved for use in recent years are derivatives of existing compounds that have been modified chemically in the laboratory. In other words, these compounds process the same core of scaffold as the compound from which they are derived, and they can be considered members of the same chemical class. Existing drugs are chemically modified and attempted to make them less vulnerable to the mechanism of resistance employed by the bacteria they target. It was hoped that the sequencing of the genomes of pathogenic bacteria beginning in 1995 would lead to the identification of host of a new drug targets, but this had